Every living thing on this planet, from the tiniest bacterium to the human body, runs on one universal code. But how does a simple sequence of letters hidden inside DNA turn into thoughts, movements, and life itself? Today, we'll trace that journey, from the elegant structure of DNA, to the script of RNA, and finally, to the proteins that build and power every cell. At the heart of biology lies one simple principle: information flows in a single direction. DNA stores the instructions, RNA carries the message, and proteins do the work. This is what scientists call the central dogma of molecular biology, the fundamental rulebook of life. To understand it, we need to begin where it all starts: the structure of DNA itself. DNA, the molecule that carries the instructions of life. It sits inside the nucleus, built as a double helix of repeating nucleotides, storing information in the form of genes. These genes decide who we are, and pass our traits to the next generation. But here's the real question: did scientists always know DNA was the hereditary molecule? Or did this discovery take years of mystery and experiments? The mystery of DNA's structure finally reached its climax in 1953. Building on decades of work, two young scientists at Cambridge University, James Watson and Francis Crick, pieced together the puzzle. Their breakthrough was inspired by critical X-ray diffraction data from Rosalind Franklin, which hinted at a spiral shape. Watson and Crick proposed that DNA is not just any spiral, it is a double helix, like a twisted ladder. Here's what their model revealed. DNA is made of two polynucleotide chains running in opposite directions, one 3 to 5 end and the other 5 to 3 ends. This antiparallel arrangement is essential for stability and replication. Bases face inward and always pair in a strict way. Adenine pairs with thymine via 2 hydrogen bonds and guanine pairs with cytosine via 3 hydrogen bonds. This explained Chargaff's rule, why adenine is equal to thymine and guanine is equal to cytosine in every DNA molecule. Pairing a large purine with a small pyrimidine keeps the DNA's diameter constant at 2 nanometers. This solved the problem of structural consistency. The bases are flat and stacked 0.34 nanometers apart, like steps of a staircase. The helix makes a full turn every 3.4 nanometers, containing 10 base pairs per turn. The double helix is stabilized not just by hydrogen bonds, but also by hydrophobic interactions and base stacking. Because of specific base pairing, DNA can be copied exactly. Each strand serves as a template for the other, the very basis of heredity. So, this is how scientists uncovered DNA over decades of research. Now let me quickly summarize it for you. DNA is a double helix, made of two long strands twisted around each other like a spiral staircase. These strands run in opposite directions, which scientists call antiparallel. And what holds them together? Nucleotides, the building blocks of DNA. A nucleotide has 3 parts: a sugar, a phosphate group, and a nitrogenous base. It's the nitrogenous bases that give DNA its code, and they come in 2 groups, purines and pyrimidines. Purines are the larger bases, with 2 rings. These are adenine and guanine. Pyrimidines are smaller, with a single ring. These are cytosine and thymine. And here's the magic. Each base pairs specifically with its partner through hydrogen bonds. Guanine pairs with cytosine, and adenine pairs with thymine. This precise pairing keeps the DNA strands tightly bound, giving the helix a consistent 2 nanometer diameter. Now, the phosphate and hydroxyl groups on the nucleotides can react chemically. When the phosphate group of one nucleotide bonds with the hydroxyl group of another, a water molecule is released, forming a strong covalent bond called a phosphodiester bond. This links nucleotides into a long chain, with a free phosphate at one end and a free hydroxyl at the other, ready to connect with more nucleotides. Thousands of nucleotides can stack on top of each other, a process called base stacking, which adds even more stability to the DNA molecule. Each base is 0.34 nanometers apart, and 10 bases complete one turn of the helix, about 3.4 nanometers in length. As the strands twist, they form two grooves, a major groove and a minor groove, which are important for protein interactions and gene regulation. And you know what the most amazing part is? DNA can be up to 2 meters long in a single cell, yet it fits neatly inside the nucleus. How? The double helix coils around proteins, forming chromatin, which then folds tightly to fit inside the tiny space of the nucleus. And that, in a nutshell, is the structure of DNA, elegant, stable, and ready to store all the information that makes you, you. Fascinating, isn't it? Have you ever wondered why scientists keep saying, DNA is the code of life? It sounds fascinating, but also mysterious. How can something so small, a thin thread-like structure, decide your skin tone, your hair color, even how tall you might grow? The secret lies in two powerful processes, transcription and translation. Think of DNA as the master blueprint of your body. But blueprints can't build a house on their own, they need workers. That's where transcription comes in. In this step, DNA makes a working copy of its instructions, called messenger RNA, or mRNA. Then, ribosomes, the cell's protein factories, step in for translation. They read the mRNA code and build proteins according to the instructions. And here's the key: proteins are what actually give you your traits. For example, proteins that produce melanin control your skin and hair color. Structural proteins shape your body, enzymes power your metabolism, and hormones regulate your growth. So, when we say DNA codes for life, what we really mean is, DNA writes the instructions, transcription makes a copy, and translation brings those instructions to life, one protein at a time. When we put all of this together, DNA making RNA, and RNA making proteins, the entire flow of information is called the central dogma of molecular biology. It's the guiding principle that explains how your genetic code becomes you. So let's dive into the first step, transcription. Transcription is like photocopying a page from the master blueprint. But there is an issue. DNA stays safely inside the nucleus, and the cell still needs its instructions outside, in the cytoplasm. To solve this, the cell makes a copy of the gene it needs, not the whole DNA, just the relevant section. Let's understand this process in three main steps: initiation, elongation, and termination. Our first step is initiation. Initiation, the first step of transcription. This is where the process actually begins. But what do we need for it to happen? Three main things: DNA, the blueprint, from which the gene has to be copied; RNA polymerase, the enzyme that does the copying; and nucleotides, the building blocks for the new RNA strand. Now, remember, DNA is a double helix, made of two strands. But only one strand is transcribed. This is called the template strand, or the antisense strand. The opposite one is the coding strand, or the sense strand. Here's a question for you. If the coding strand already looks like the RNA we want, then why does RNA polymerase copy the opposite strand instead? Think about it. I'll reveal the reason in a moment. First, let's see how RNA polymerase gets started. Transcription always occurs in the 5-3 direction. It begins at a specific site on the DNA called the promoter, a short sequence, usually about 50 base pairs, located near the transcription start site. The promoter is essential because it tells RNA polymerase exactly where to begin. A classic feature of many promoters is the Tata box, found about 25 to 35 bases upstream from the start site. In eukaryotic cells, initiation begins when general transcription factors and RNA polymerase II bind to the promoter, forming what's called a pre-initiation complex. Once this complex is assembled, the DNA strands are separated at the start site, and RNA polymerase is positioned perfectly to begin synthesizing mRNA. And with that, the stage is set for the next step, elongation. Elongation, the second step of transcription. This is where the real action begins, the actual synthesis of mRNA. Once the transcription factors have done their job and correctly positioned RNA polymerase at the promoter, RNA polymerase moves forward along the template strand of DNA. Here's how it works. The DNA double helix unwinds just ahead of the polymerase, opening up a small transcription bubble. Inside this bubble, RNA polymerase reads the DNA template strand one base at a time. Complementary RNA nucleotides are added, forming the growing mRNA chain. But here's something important. Unlike DNA, RNA doesn't contain thymine. Instead, it uses uracil. So whenever the DNA template has adenine, RNA polymerase adds uracil in its place. As elongation continues, the mRNA strand does not remain bound to the DNA template. Instead, it peels away, while the DNA helix immediately zips back together behind the moving enzyme. RNA polymerase keeps sliding along the template, building the mRNA strand in the 5-3 direction, nucleotide by nucleotide. In eukaryotes, this process can create transcripts thousands of bases long. Think of it like a typewriter that never stops, moving forward, unzipping, copying, and sealing the DNA behind it, while producing a fresh ribbon of RNA. And with elongation in full swing, we're now heading toward the final stage of transcription, termination. Once RNA polymerase II finishes copying the gene, it doesn't stop immediately. Instead, it keeps moving forward, transcribing extra bases beyond the coding region, almost like a typewriter that keeps pressing keys even after the sentence has ended. But within this extra stretch lies an important signal, a short sequence in the RNA called the polyadenylation signal, and it tells the cell, the useful part of the transcript ends around here. At this stage, proteins called cleavage factors step in. They recognize the signal and cut the RNA strand just a little downstream. This separates the fresh RNA transcript from the polymerase. Meanwhile, RNA polymerase II continues along the DNA for a short distance, producing extra RNA that has no real function. Eventually, it falls off the DNA, and the double helix snaps back into place. Remember we asked why RNA copies the template strand instead of the coding strand. Now you'll see, by copying the opposite strand, the RNA ends up with the same sequence as the coding strand, except with U instead of T. In other words, the coding strand represents the gene, but the template strand is what makes transcription possible. Now comes the finishing touch. An enzyme called polymerase adds a long tail made of adenine bases, about 100 to 250 of them. This polymer tail protects the RNA, helps it leave the nucleus, and prepares it for translation. The result is a brand new pre-mRNA, essentially the first draft of the gene. But before it can be used, this draft needs editing. Through post-transcriptional modifications, the pre-mRNA is capped, spliced, and fitted with a poly-A tail. Only then does it become a mature mRNA molecule, ready to exit the nucleus and guide the process of protein synthesis. This mRNA now leaves the safety of the nucleus through tiny gateways in the nuclear membrane called nuclear pores. Once in the cytoplasm, the message is ready for the next big step: translation. Translation is the biological process in which the sequence of nucleotides in messenger RNA is decoded to assemble a chain of amino acids, forming a specific protein. In simpler words, it's the step where the genetic code written in mRNA is translated into a functional protein. It is the second step of the central dogma and takes place in the cytoplasm. For translation to happen, the cell requires mRNA, carrying the genetic message from the nucleus. Ribosomal subunits, the protein-building machines. tRNA molecules, which bring in the right amino acids, and the amino acids themselves, the raw material of proteins. It's actually a simple yet fascinating process, almost like a well-coordinated game. And this game unfolds in three main stages: initiation, elongation, and termination. Initiation is the starting point of protein synthesis. Here's how it unfolds. The small ribosomal subunit attaches to the mRNA strand at a specific site, close to the start codon. A codon is simply a group of 3 nucleotides that codes for one amino acid. In this case, AUG is the universal start signal. A transfer RNA carrying the amino acid methionine, the universal start amino acid, recognizes the AUG codon and pairs with it using its anticodon region. An anticodon is a set of 3 bases on the transfer RNA, complementary to the codon on the mRNA. This ensures accuracy, because each codon in the mRNA has a matching anticodon on a specific tRNA, and every tRNA carries only its designated amino acid. The large ribosomal subunit then joins the complex, forming a complete ribosome. This structure now has 3 important sites that control the flow of translation. First, a site or aminoacyl site, the entry point where each new transfer RNA carrying an amino acid arrives. Second, P site or peptidal site, the spot where the growing polypeptide chain is held and new bonds are formed. And the third, E site or exit site, the exit door where empty transfer RNA molecules leave after delivering their amino acid. With the ribosome fully assembled and the first amino acid in place at the P site, the protein building machinery is ready to move on to the next stage, elongation, where the chain starts growing one amino acid at a time. Once the initiation complex is ready, the ribosome begins the actual process of building the protein chain. Here's how elongation works step by step. After the start codon is set at the P site, the ribosome reads the next codon on the mRNA. A tRNA carrying the corresponding amino acid enters the A site and pairs its anticodon with the codon on the mRNA. The amino acid at the P site that is methionine is joined to the new amino acid at the A site by a peptide bond. This reaction is catalyzed by the ribosome's enzymatic activity, specifically the peptide L transferase function. This is how a peptide bond forms. One amino acid has a carboxyl group, and the next amino acid has an amino group. During the reaction, the hydroxyl group from the carboxyl end of one amino acid and a hydrogen atom from the amino group of the next amino acid are removed. Together, they form a molecule of water. What's left behind is a new bond between the carbon of the carboxyl group and the nitrogen of the amino group. This bond is called a peptide bond. In short, peptide bond formation is a dehydration reaction that links amino acids into a growing polypeptide chain. Now, the ribosome shifts one codon forward along the mRNA. The tRNA that was in the P site now moves to the E site and exits. The tRNA that was in the A site, with the growing peptide chain, shifts into the P site. The A site is now empty, ready for the next incoming tRNA. This cycle, codon recognition, peptide bond formation, and translocation, repeats over and over, causing the polypeptide chain to grow longer with each round. Termination is the stage where protein synthesis comes to an end. Here's how it happens. As the ribosome moves along the mRNA, eventually it reaches a stop codon. A stop codon may be one of the 3. Unlike other codons, these do not code for any amino acid. Instead, they signal the ribosome that the protein is complete. A special protein called the release factor binds to the stop codon in the A site of the ribosome. This triggers the ribosome to cut the bond between the polypeptide chain and the final tRNA in the P site. The newly made polypeptide chain is released into the cytoplasm, ready to fold into its unique 3D structure. The ribosomal subunits, tRNA, and mRNA then separate and can be recycled for another round of translation. And that's the complete journey of protein synthesis: initiation, elongation, and termination. From a simple sequence of nucleotides in mRNA, the cell constructs a full polypeptide chain, the foundation of proteins. Later, this chain undergoes folding and post-translational modifications, transforming into a functional protein that carries out essential tasks in the body. But our story doesn't end here. The product of translation is a polypeptide chain, and on its own, it's not yet functional. To become a true protein, this chain folds into a specific 3D shape, guided by various chemical interactions and bonds, such as hydrogen bonds, ionic bonds, and disulfide bridges. It's this precise folding that gives every protein its unique shape and, ultimately, its unique function. Once properly folded, the protein may stay inside the cell to perform its role, or it may be transported out of the cell to work elsewhere in the body, like enzymes in digestion, antibodies in immunity, or hormones in signaling. And so, the journey of translation closes, a process where molecular precision turns genetic information into the very proteins that power life. Each protein crafted is unique, purposeful, and vital, silently shaping everything from the strength of our muscles to the sharpness of our thoughts. It's a reminder that even the tiniest processes inside our cells carry the blueprint of life's complexity and beauty.